Good afternoon, good evening, good morning, everybody. Welcome once again to week two, and actually week three. Sorry, welcome to week three. I messed that up of the development and analysis of human movement. Today we are going to go over muscular considerations. Because this is a relatively dense chapter, it does have some key concepts in it that I do want to make sure we really understand. I am going to break it up into two separate videos this week. I'm going to post one tonight, one tomorrow. That way we have both of them and it kind of breaks it up for you guys. So you don't all have to sit there for close to an hour and a half, 2 hours listening to one video. Okay, I know you can pause it, but it's just easier if I break it into two. So let's get started. Our objectives this afternoon is to understand to gain knowledge in the anatomy of bone, skeletal, muscle, and connective tissues, to gain knowledge in the muscle action terms, including anterior, posterior, inferior, superior, medial, lateral, supination, pronation, flexion, extension, abduction, adduction, hyperextension, rotation, circumduction, agonist, antagonist, and stabilizer. Today we're really going to work on agonists and antagonists along with stabilizers while reinforcing some of those other terms that we learned the first week of class. You're going to gain a knowledge of the 6 motor skill related physical fitness concepts: agility, balance, coordination, reaction time, speed, and power. Most specifically, we're going to focus on a little bit of speed and a little bit of power as musculoskeletal, not musculoskeletal, but muscle attributes to those. So we're going to kind of touch on them just a little bit. We're going to gain some knowledge in the joint movements: flexion, extension, abduction, adduction, and the muscles responsible for them. So we're just going to touch on it. We're still not diving into the specifics, but we are going to just kind of touch it. So I put it in there. We're also going to gain a knowledge of the anatomy and physiology of skeletal muscle fiber, the characteristics of fast and slow twitch muscle fibers, the sliding filament theory of muscle contraction, and we are finally going to gain a knowledge in the normal maturational changes from childhood to old age and their effects on the skeletal muscle, bone, reaction time, coordination, posture, heat and cold tolerance, maximal oxygen consumption, strength, flexibility, body composition, resting, and maximal heart rate, and resting and maximal blood pressure. Really, we're just going to really focus on the first part of that, normal maturational changes from adult to childhood. Okay, we're not, and their effects on skeletal muscle, but not so much the rest. So, as I said earlier, what we're really diving into this week is muscle tissue. So, to understand muscle tissue, we need to understand some of the properties that muscle tissue have to give them the characteristics that they carry. So, one of them is irritability. While you might think of irritability as what most people in these online classes do to you while you're sitting in your room, it is, in terms of muscle, it is the ability to respond to a stimulus. So, if a muscle is irritable, it has the ability to respond to some type of stimulus. Often, during that, a motor neuron sends a neurotransmitter as that stimulus within our bodies. Only nervous tissue is more sensitive than muscle as it relates to irritability. Okay? And muscle tissue is highly sensitive, and it allows muscle to be recruited very quickly and also very specifically in a controlled manner. So once it is irritated, it gains a contractility, and that is the ability to shorten when stimulated. Most muscles within the body are able to shorten from their original resting position about 57%, but it could be as high as 70% in the total length. It's mostly just limited by other muscle tissue, other skeletal tissue around there, such as bone or other, like your psoas muscle within your low back and your hip. That becomes limited in how much it can actually shorten by the bone hitting, the femur hitting the ilium or the iliac crest as it goes into hip flexion. Let me show you here. It just kind of gives you a little idea. Wrong button. Turns it off. So like as you look here, you can see that there's muscle that is attached here, right? And I said the psoas muscle. So let's go ahead and hide that, and let's get underneath here. Get rid of that. Let's get rid of this guy, this guy, this guy, and this guy. Yeah, why not? Since we're just cutting everything off, huh? Okay. So you can see here that where this muscle aligns in the body, or as it originates, as you'll learn later, is what it's called. On the spine here, we have L5, 4, 3, 2, 1. It goes all the way up to here. It runs down, right? This is the psoas muscle, as I said, right? Psoas major, okay? And what it does is it comes down and it attaches to the femur. But if I was to shorten this, what would happen, right? This muscle would come in and it would also pull on our back. But eventually this femur will hit this bone here, right here. Okay. So as it hits that iliac, that ilium, we call it the iliac crest right here, and that's where it would hit. Boom, it would hit the iliac crest and thus cause a limited range of motion. Thus it could probably shorten if we cut it to 50% all the way up to here. But because that bone kind of hits itself there, along with, if you look at the other side where the muscle mass is still there, you can look here at this vastus lateralis right here, and look how much muscle tissue is on top of that bone. So this muscle tissue is also going to stop it from going all the way up. So other tissues will also have that happen. Another property of muscle is its ability to be extensible. So what that means is its ability to lengthen. So an external force is required for elongation, and connective tissue determines the amount of extensibility. So if we go back to our model, you look here, right? So elongation, it needs to be able to elongate, it says, right? Especially as you look here. So if I'm going to flex my hip and bring it up close, right? As I pull it up here, all the anterior muscles are going to shorten. Right? So all these muscles up here are going to shrink and shorten, but all the muscles on the back, if I'm lifting my leg up this way, right, they need to elongate, okay? And that's really limited by these tendons down here. How much these tendons contract and the properties of those tendons is what will limit some of that elongation capability within that tissue. And then finally, they need to be elastic. The elasticity of a muscle tissue is really the ability of that muscle to return to its resting position once it's unloaded. So that's why you think of it, or I think of it, as a rubber band. And why when we get into lab, we're actually going to use our rubber bands to represent muscles, because muscles and rubber bands have very similar properties, right? A rubber band needs to respond to a stimulus. It needs to be able to stretch and hold that muscle or hold that tension fast in a controlled manner. It needs to be able to shorten when it's stimulated, right? So it can shrink a little bit. And then for our purpose, it needs to be extensible. It needs to be able to lengthen. And then when we remove that load, it needs to be elastic and be able to shorten and go back to its original resting position. Okay? Again, the connective tissue really plays a factor in this ability. So as we look at some of the functions of muscle tissue, we have to look at what is its job. So the main job of muscle tissue is to produce a movement. As the muscle contracts, a force is transferred to the bone. So you have to imagine it, not imagine, let's look at it, right? So the muscle attaches to bone, right? All these muscles here are attaching to bone in some various capacity. So whether it's from here to here, right, into our knee, into our patellar tendon, as we come from our ASIS here, all the way down into the tibia down here, whether it's in our forearm, right, and our palmaris longus as it just works down and it works to flex our hand, right? No matter what, it's attaching from one bone to another bone. And so as a contraction, it needs to transfer this force efficiently via the tendons into the bone. It needs to be able to maintain posture. So the irritability of muscle tissue, if we go back, right, we said it has the ability to respond to a stimulus and that it's highly sensitive and it allows the muscle to be recruited fast and in a controlled manner. So that controlled matter allows us to have minor contractions throughout the day as we just sit, as I'm sitting in this chair here. It's allowing me to make slight modifications to maintain my position, whether I move anterior or posterior or lateral, right? It allows me to maintain those very subtly, so much that it looks like I'm sitting still, but really I'm very dynamic as my body's doing these small little thrusters and contractions trying to maintain my desired position. Another thing that it does is really stabilizes joints. Ligaments allow for forces to cross the joints and stabilize them. So one great example of that is if we move from here and we move up to the shoulder, right? So we look at the shoulder here and it's very stable, right? And why is that? Because of all this muscle tissue. So, sorry, turn us here. Sorry. There we go. So we look at all this muscle tissue. So we take off the deltoids, and now what do we have, right? Don't have much left. I'm going to remove the traps here, and let's take a look. What's left? Around that humerus, you don't see much, but you do see a lot of white, don't you? A lot of white. And that's all the rotator cuff muscles. I'm going to remove one more rotator cuff muscle right here. And you can really see that these tendons cross the joint so that we don't have this massive amount of tissue crossing the joint. I can still get stabilization from this biceps tendon right here as it crosses that humerus. And from the, there we go, supraspinatus and the infraspinatus, those tendons are allowed to cross over now. And our teres right here, those all cross over to help stabilize this humerus and its acetabulum here, and its little fossa. So that's what we mean by it. Muscles help stabilize the joints via the tendons, okay? Other abilities are protecting organs and tissues, controlling pressure, temperature control, and they control entrance and exits of the body, right? So protects organs and tissues. What do we mean there, right? So let's look here. Let's just use this spot here. I don't want to reset the whole model. So if we look here, right, all our organs would sit inside here. And you have this thick aponeurosis right here of the rectus obliques and the rectus abdominis underneath there as we remove those, and those help protect all of our organs. So now we'll move on to pressure. Got my light. So the diaphragm right here, underneath here, you can kind of see, there we go, right there at the diaphragm, controls abdominal pressure and interthecal pressure, and it massages all the organs that sit in here. So they help with respiration, digestion, and overall blood flow and pressure within the abdominal cavity. Temperature control. Muscles control temperature, body temperature through involuntary shivering and heating up through contracting and elongating by increasing blood flow to the area, and that's how that happens. And then it controls the entrance and exits of the body. So you can see here the mouth, how it's a circular shape, muscle here that opens and closes around the eyes, the same, right? You can see that, how it would control those openings and closings, okay? So, organization. Muscles are arranged so that muscles can fire individually or collectively. They're bound together by what we call fascia, and often a group is innervated by the same nerve. And a compartment syndrome can be a problem. So if we look at our diagram over here on the left, this is a picture of the lower leg down here, okay? And if you look here, you have different compartments. You have the anterior compartment over here, you have the medial compartment in here, and you have the posterior compartment back here. And so the posterior compartment on the thigh, the upper portion, consists of like the hamstring muscles. Then your medial compartment is mostly your adductors, your adductor magnus, adductor longus, sartorius, gracilis. Then over here, the anterior, you have vastus lateralis, rectus femoris, vastus medialis, and then you can see how each individual has its own little fascia binding connecting all the muscle fibers, and then all together it's kind of bound into its own little compartment as well. You can see it even more prominent in the lower leg, where you have the posterior compartment with your gastrocs and soleus here. You have your deep posterior compartment with your toe flexors and tibialis posterior in here. And then you have your anterior compartment and your lateral compartment over here. Compartment syndrome can be a problem when a muscle in these compartments gets a little too hypertrophied and causes too much pressure and constricts the blood flow or the nerve endings in its individualized compartment. So now we get into the different type of muscles, so the anatomy of the muscles. Okay, we have parallel muscles and peniform muscles. Parallel muscles are is where the fascicles are parallel to the long axis of the muscles. They originate from an aponeurosis, which is a long sheet-like type muscle. They're different shapes that allow for focused force application, and the fibers run longer than the tendon typically, and they really are most applicable for creating a large range of motion, and they move it fast, really fast. Then you have peniform muscles, which are like feathers, you could almost think. And the fibers run diagonal to the central tendon, and you get typically less range of motion out of those, and they're a little bit more slow, but you get more power because there's a larger cross section of those. So you can see here in A, you get a parallel muscle where the muscle runs in the same line of pull here as the tendon. So tendon here at the top, tendon at the bottom, and then the muscle fibers, you see the lines are arranged parallel, right in line. But then you have these peniform muscles here, like your gastroc. If you think of that like horseshoe shape that a lot of people look for in their calves, that's this bipennate gastroc right here, where you have this long tendon in the middle, and the line of pull is here, but then you have the medial and lateral gastroc, and the fibers run almost perpendicular there. They run diagonal, kind of about 45 degrees from that line of pull, from that tendon. So you can see how that pennate. Then you have unipennate, where it's just one muscle here. This is the semimembranosus, where it pulls just one. Bi, where it's pulling two different directions. Then you have a multipennate, which is like a deltoid, where it runs into one central tendon, and you have this almost fascial, like musculotendinous piece that runs into the muscle belly and divides the muscle belly so that it moves in multi-directions. It's almost like a layered bipennate type muscle. Now when we're looking at pennation muscles, you have these pennation angles, okay? And the angle of the muscle fascicles and the line of pull is what you really want to look at. The wider the angle, the less force transmitted to the tendon. And then as the muscle contracts, the angle widens. So if we look back here, right, if we think of this muscle contracting, as it contracts, this is going to shorten. This whole muscle tissue right here, in this bipennate gastroc, is going to shorten, so it's going to get smaller. So if I go up and bring this white piece up, what's going to happen to these lines? They're going to move out like that, okay? And then the wider that angle, the less pull. So as it goes out, it's going to have less force produced than if it was more of a sharp, smaller angle, a more steep angle, I guess you could say. Okay. Then you have muscle volume. Muscle volume is very critical to determining the amount of force that a muscle can produce. So muscle volume, the equation is muscle volume equals mass times volume. Okay. And that gives you your muscle volume. Physiological cross-section is what you'll look at a lot. So physiological cross-section is what this is here, and that's the mass times cosine of the angle times the change in length times the volume. And that is directly proportional to the amount of force production available. So mostly muscle volume in general, so like how big the muscle is great. But what you really want to look at is the physiological cross-section, because that's what's going to be more indicative of how much force can be produced by a muscle. So then we have muscle fibers. I'm sure you guys have heard of by now fast twitch and slow twitch muscle fibers. So type 1 muscle fibers are slow twitch fibers. They have a high amount of myoglobin in them, which means they have a lot of oxygen within them. So those are most utilized during prolonged low intensity work. That's where you think of your endurance runner, your endurance athlete. Then you have type 2, which are fast twitch muscle fibers, and then those are subdivided into 2A and 2B. 2A is an oxidative glycolytic muscle fiber. It sustains prolonged activity and short bursts, and then they fatigue. So you can sustain this longer duration, but it's still just more of a short burst, and it fatigues off. Then your type 2B are your glycolytic. These are your sprinter type muscles here, where you get a very rapid force production and a very, very quick burn off of those muscle fibers. So when we look at the actual anatomy of a muscle, what are we looking at? So we have the muscle belly. That's what we all think of when we look at our human person, right? This nice red stuff, that's our muscle belly right there. And you have your epimysium, which is the fibrous covering of the muscle. So you then move down even lower. So if you think of this as going from top to bottom, right? Then you have the fascicles, which is individual fibers that are bundled together. And some muscles, they may contain up to 200 fibers within each little fascicle. Then each fascicle is covered in a perimysium, which provides a path for the nerves and blood supply to move within. And this is where you would see some of those issues with those compartment syndromes when the fascicles get too large and hypertrophied. They kind of stress that perimysium and cause a contraction and a blockage within the little channels within there. Then you have the fibers. So once you have your fascicles and your perimysium that connects that turns into the fibers, and those are long cylindrical thread-like. Sorry, reverse what I just said. So the fibers make up the fascicles, are bound in perimysium, which make the epimysium, and then the muscle belly. Okay, so your fibers are the most basic part down there, and those are the long cylindrical thread-like tissues that run parallel to each other. Typically, those are measured in meters, and that runs from 10 to 100 centimeters in length and 15 to 30 centimeters in width. So if we look over here at our picture here, it shows how you have this muscle belly here, and then it's broken down, right? You have your muscle belly, then you can see the epimysium in there. And then that's broken down. If you look at each one of these little blue circles, it takes one, it expounds it here, and now you have this fascicle here, which is then tied in with little epimysium in there, and then the perimysium around each little bundle, right, which gives you your muscle fiber. And then you have your sarcolemma is your sarcoplasms, your myofibrils, and then your sarcomeres. Okay, we're going to dive into those in a second here. Right here, see? Your endomysium. So what is the endomysium? That is right here, right? Your endomysium, your epimysium here. The endomysium is the membrane that covers each individual fiber, and it carries capillaries and nerves. So pretty much if you look, like, you get some type of ecium that's like your wrapping, right? It's like a saran wrap that holds everything together. So you have your muscle belly, then you have your epimysium, which kind of covers that. Your fascicles, which is covered by your perimysium. Your fibers, which is covered by your endomysium. Your sarcolemma, and then your myofibrils. Okay? So your sarcolemma is the plasma membrane that nerve innervation travels via neurotransmitter. We'll dive into that in just a minute, a little bit deeper. And then your myofibrils, which are rod-like structures that contain the contractile proteins of the muscle that allow it to actually contract. And that's what fills up about 80% of those actual muscle fibers. Okay, so you can see these myofibrils and these sarcomeres, they kind of alternate between each one. So you see how it's a little lighter, dark, light, dark, light, dark. So it's almost like an alternating chain of myofibrils and sarcomeres. And then when you look even lower, you can see the bands here. The thick bands here are the myosin that sit within there. And then you have this thin filament, which is like your actin, and that's the black lines here. So as we move forward, you'll really get to understand that. So how do muscles actually generate the force that we ask them to provide? Well, it typically starts at the motor unit. The motor unit is a group of muscle fibers innervated by the same motor neuron. It can go from a few up to 2,000 muscle fibers in each motor unit. So that you can see how that can be very specific or very broad in how each motor unit stimulates the amount of muscle fibers. Of course, that's directly proportional to how much force a muscle can generate. If a motor unit activates just a few muscle fibers versus 2,000, I'm going to definitely get much more force out of 2,000 muscle fibers than I will just a few. So then you have an action potential, which is what we call the signal from the neuron to the muscle initiating a contraction. So once the muscle receives an action potential from the neuron, that's when you start to get that contraction. When that signal is received, all fibers are innervated by that motor unit. That's when they contract. And the size of that action potential is proportional to the number of fibers in the motor unit. So like I said here, if we have a few, we're not going to get a very large force, right? We're not going to get a very large action potential or contraction as we would if we had an action potential on 2,000 muscle fibers. If I have a larger action potential, I'm going to get a larger force by activating 2,000 than I would if I activated just a few. So what actually happens when we get this action potential from this neurotransmitter, right? The action potential rides at this neuromuscular junction within each muscle fiber. In that muscle fiber, there's a synapse between a motor neuron and a fiber, so a little gap. What happens is that acetylcholine crosses that synapse, and it increases the permeability of the sarcolemma, and it breaks down real quick. Otherwise, it leaves that open, and then that's where you start getting too much of a action potential. So once that happens, once that permeability of the sarcolemma is provided, then calcium enters the sarcolemma and it surrounds the myofibrils. This starts a cross bridging, which leads to an interaction between the myosin and the actin. And that's where we start really getting into what's called the sliding filament theory. It's the most widely accepted explanation of a muscle contraction, what's actually happening physiologically when we get that. And this is when that calcium is released, that contraction really occurs. So once that calcium enters the sarcolemma and surrounds these myofibrils, and we get the cross-bridging action of the myosin and actin, and that's where we get that contraction. The sarcomere contracts as myosin slides along the actin, and when it is contracted, the myosin and the actin overlap a majority of the length, so they cross over. We have a good picture here coming up, and you'll be able to see. This microscopic interaction creates a shortening of the entire muscle, and this is once again proportional to the number of cross bridges formed. So that it's, before we were saying the action potential is directly proportional to the amount of fibers, and then at a microscopic level, we'd look even deeper, and you see that the amount of force is proportional to the number of cross bridges. So the more cross bridges I have, the more force I'm able to apply, because I'm activating more muscle fibers to contract. And then if we look back, the more fibers that are contracted, the greater force I'm allowed, or not allowed, the greater force I'm able to produce in the actual muscle. So if we start on the left here, you can really see the picture of that sliding filament theory. So what we have is the myofibril and the sarcomere, right? And then you look a little bit, it does a cross section and it kind of cuts that out. And now you get this bare zone, and you see your actin here at the top, the nice little, it almost looks like a pipe cleaner, right? Then you have your myosin down here that almost looks like a piece of asparagus, right? So if we look closer, what we see is in this actin and in this myosin, you see the actin here and then the myosin here, okay? So what happens is this actin has this little appendage that reaches out, and it's looking for a spot within this myosin. Once it finds it, it attaches on, and it bends. You see how the arm kind of bends and pulls this actin? So you can almost think of it as pulling a link in a chain like this, right? And it finds it, pulls it, and then it detaches and finds the next spot and pulls it. So as it happens, that's how the muscle contracts, right there. So then this is a larger picture from the top here. You can see the acetylcholine is released, binding to the receptors, right, within our body. Then the actual action potential reaches the T-tubule, right here. And then we have this sarcoplasmic reticulum that releases the calcium ions. You see that right here, right? And then 4, the active site exposure creates a cross-bridge binding, and that's what we just talked about over here. And you can see there the calcium, the actin, and you see the myosin pulling on them, right? And then you see here the contraction begins, and that's how you start getting this pulling right here. Then after that, the acetylcholine is removed. And then you get the sarcoplasmic reticulum recapturing. It loses that permeability and it pulls back that calcium. Then the active sites are corrected, and there's no more cross-bridging. They're not reaching, they're not connecting anymore. And then our contraction ends. So it's no longer covered up. It stopped, it pulled it back. It's not permeable, so the sarcoplasma has lost that, and it's no longer receiving those neurotransmitters that connect that. So then we move on, and we start looking at how this force is directed to the bone. So when we look at that, we look at it's either through an epimysium, a tendon, or aponeurosis. Okay, an epimysium, it fuses with the bone at the periosteum. So it's a direct link. So if you look at picture A here, like our coracobrachialis, it almost just, the muscle just goes straight into the bone. And while it looks like it has, there's actually a very small junction in there where this epimysium is fused with the bone. And in the muscle almost, there's a little bit of mineralization and calcification that kind of occurs where the bone and the muscle start becoming one and attaching into that. Then you have the most common way, which is the tendon. Then the tendon that attaches the muscle to the bone so that it can apply the force of the muscle to the bone in a lever-like system and a thick connective tissue band called a tendon. The muscle fascia and tendon attached to the bone, right down here, in the same type of protuberance or tubercle that we were talking about before in the skeletal system, where those bony outgrowths kind of occur. And you can see that here, right? You can see how a lot of tendons kind of attach on the outer part of this humerus here. And if I take them away, you can see that the body has kind of grown this little groove and this little projection tubercle here so that all those tendons can attach there. And then we have an aponeurosis, which is a sheath of fibrous tissue. So I don't like the picture in this one, so we can look down here. So like this white piece here, this would be like an aponeurosis. And you see how the tissue just kind of, it's flat tissue here in these oblique muscles. And it just kind of attaches to this big connective band right here. And this is how you start getting hernias and stuff. When this band of connective tissue tears, then the muscles underlying start protruding out, and that's where you get these hernias. But yeah, you can see how broad this piece of fascial connective tissue is compared to the muscle belly, compared to a tendon. So look here in the arm, how this tendon's nice and right here. This tendon's very thin, very focused in where it needs to go, down into the hand, and it connects to the bone down here. And even down here, you can see that there's little protuberances here within the bone to handle the force that it's pulling. Whereas here, it's a broad connective tissue, and it's the same on the other side. We just took those layers off to look inside. Okay. So tendons. If we look at tendons themselves, they're the most common form of attachment within the body, from muscle to bone. They create in the muscle this muscular tendinous unit, and that's where the muscle, you see here, that this tendon starts turning a little bit broader, going from that focal cord into a broad piece, and then it starts becoming a little bit less connective tissue and a little bit more muscle, and that's what we call the myotendinous junction. It's typically where the muscle begins to be woven with a little bit of collagen. That's what makes up most of that tendon, and it's that kind of a nice blend of that. Typically, tendons are circular, oval, or flat. They resist stretch, they're flexible, and they can turn corners. And that's important as we start looking at leverage and how those things occur. Because you look down here on our ankles, right? Look how they turn as they move around our lateral malleolus here, as the tendons move around our calcaneus and our Achilles, right? Our deep toe extensors here in the anterior compartment, they bend around this bone, around the talus, and they kind of connect on these toes. But you see how they're flexible, they're round. You look at the ankle and you see how they're round here, a little more flat as they move down here. So very different in shape depending on their desired function. They have a stress-strain response that allow them to be stiff so that they can form the muscle, or the force from the muscle can be transmitted to the bone in the correct amount, and you lose very little energy. So if they're real elastic, what would happen is as that muscle contracted, the tendon would contract as well, and now it doesn't work. So imagine trying to pull a 45-pound plate on the ground with a rubber band. What's going to happen if you attach a rubber band to that plate and then you pull on it? You get a stretch. You're going to lose a lot of your force in that rubber band as it pulls apart. So that's why you want something a little bit more stiff. But if it was too stiff, if it was, let's say, like a piece of glass, as you pull on it, it may just break in half. So it very much has the stress-strain response that allows it just enough stiffness to handle the force that the muscle's asking without breaking, and also enough stiffness that it doesn't stretch so much that you lose all your force and the muscle becomes inefficient. At the bone, the fibrocartilage joins a mineralized fibrocartilage to merge with the periosteum and the subchondral bone. tendons bear the same tension as muscles. So force, speed, and laxity determine the mechanics of the tendon. Okay, because they have that same tension as the muscle. So as the muscle contracts, the same tension that's in the muscle is in the tendon. So as we look at the tendon during loading, what happens is that when the muscle is originally stretched, the tendon initially stretches and then it recoils. So you have this quick recoil response that happens in the tendon. So as soon as you pull on it, it stretches, but then it starts contracting a little bit. So as it kind of goes back to that elastic property that we were talking about earlier that it has, where it stretches and allows some stretch, but if we had, just like with the rubber band with the plate, if it stretched too much, we'd lose all our force. So it stretches and then it starts coming back. As it starts coming back, what happens is you lose some of that speed of the muscle contraction. And then as we lose the speed of that muscle contraction, it increases the load support that's allowed in that muscle. So the amount that that muscle and the tendon can hold is increased. And if the tendon is stiff, the load support is decreased and then the velocity is increased due to the tension being directed from the tendon back into the muscle belly. So the tendon and the muscle are susceptible to injury when they're stretched and contracting at the same time. That's what we'll learn is called like an eccentric contraction. The tendons travel over bone to change tissue tension if the muscle is not strong enough, as we'll learn later, that's where it creates this pulley lever type system to help unload the muscle but still increase the amount of force and change direction of the force being applied to the bone. So as we look at the mechanics of muscles, how they actually work mechanically in the body, we have to look at how they're attached. So at the top, or the most superior proximal attachment or place within the muscle, we call that the origin. It's the closest to the midline. It's typically the most proximal, and it is often more broad than the distal attachment. The distal attachment is what we call the insertion. It's further from midline, and it usually ends in that tendon that we just discussed. The force of a muscle is typically distributed between the origin and the insertion. Now, one side often seems to move when we're looking at a muscle and a tendon and how it actually moves a joint, but due to stabilizing muscles and mass differential, both sides are actually moving. It's just one side seems that way. Okay, so let's break down what I just said there, right? So let's look at this side because I haven't cut it apart. And let's look at the biceps, right? So when we think of the biceps contracting, everyone kind of has an idea of what we're talking about. Let me hide some stuff here. Okay. There we go. Let me get rid of up here. The deltoids. Boom. Let's get rid of our triceps too. Okay. There we go. So when we think about our biceps contracting, what are we looking at, right? It attaches down here. Our long head and short head attach on the ulna and radius, right? Which helps bend our arm. So it helps do that. When I think of flex your bicep, you're doing this, right? You think the biceps brings this part closer. But if we think of it on a strictly mechanical level, when this muscle contracts, both ends are coming in, right? So we get up here, this proximal portion comes down, this distal portion comes down, comes in, comes up. All right, so this whole thing, if I detach it, it would just go into a ball if I applied the stimulus that allowed it to contract, right? Boom, into a ball. So what stops up here? Why does our shoulder not, typically when we flex here, why does our shoulder not come up in like that? Because technically that's what should happen as I flex that. Well, let's look at the amount of mass that we have on our top versus our bottom. So let's reset. And here, you see how much muscle mass we have here at the distal portion versus this proximal portion, right? Look at all this muscle mass that we have. And this is why it doesn't come closer, especially when you look at the posterior, right? The anterior part, there's not much there. But if I look at the posterior, what keeps the shoulder from coming forward? All this posterior musculature here, holding that backwards. Look at this giant muscle here that's attached. You have 2 or 3 of them that sit there and kind of help pull that back. And these are stronger than the biceps itself, encounters the force, so that when we contract our biceps, our shoulder doesn't move, and just our ulna and radius and hand move. Okay? So hopefully that kind of clarifies what I'm talking about there. So as we look at that, what we're really doing is utilizing torque to move a joint. So muscles move bones using this type of system, and it further breaks it down into levers and you know, axes in terms of that. And when we look at what the actual definition of torque is, it's a tendency of a force to produce a rotation about a specific axis. So this force travels along a line of action. So if we're looking here at this blue line, this is mostly our line of action here, right? And that's where it's happening. And then you have a moment arm, which is a perpendicular distance between the line of action and the axis. The larger the moment arm, the larger the force. So if we look at A to B, right, A to B, down here we have a very little moment arm. So that's this little dotted line right here. See how short that is? Our line of action is still the same because it still originates up here and inserts down here, right? So in the same spots, but as I move my arm into that flexion position, so as I move from here to here, what happens is that moment arm increases, right? Because that line of action actually moves further from the axis there. So it's further apart, thus creating more force. And then the further this is, the more torque I'm able to create and the more force I'm able to apply. Okay? So torque, as we work later in the semester, you might need to know this equation as we work on our own biomechanical videos. And torque is the magnitude of a force in newtons times the distance of the axis to the line of action. So this little right here. This little here, sorry, right here. Okay? So it's the distance from the axis, the line of action, the origin of the insertion. So during muscular contraction, not all force is directed into the insertion. Some of that force goes into the rotation of bone. So we lose some of that force, right? And the force changes as we move the joint through its range of motion. So you can see here, it's kind of a little bit blurry, but if you have your book, it's on page, let me look here. 73. It's on page 73 for you. And you can see how these lines of pull change. As my arm's extended, you see the P here. And when you look down here, it helps identify that, right? The parallel component of the force is P, and it's the highest, it's stabilizing in the joint. And that's right here, right? So it's high because that radius is that insertion as it comes down, is going straight up through the joint, through the humerus. So it's pretty strong at that point. And then as we look here, as we start to flex it, it's still moving in through the humerus, but then as we hit 90 degrees, this is where it's pretty much the strongest, strong. But now, as we get it further into flexion, what happens is you get a dislocating force because now it's leaving there. It's not really going through the humerus, and now that's just driving the humerus, or the ulna and radius, it's driving it out and back. And as we get into a further flexion, all that force is just going straight out of the joint. It's not actually going up through the bones, so it just leaves the joint, and it actually makes it a little bit weaker through there. So this is just kind of explained further here, where we look at this perpendicular distance, and this is where a muscle has a small perpendicular distance when a muscle creates a stabilizing force. So all the force is generated within that muscle, it goes directly back into the joint. So it's efficient at that point. An angular force is most of the force is created by the muscle. Most of the force that is created by the muscle is directed at rotating the bone, right? So as we move through our ranges of motion here, you see how it changes, right? And then we get a dislocating force when the force applied past that 90-degree mark is directed away from the joint. So you see here how it's not, right? All the force directed right back into the joint, right back in, strong. Then it starts rotating here. So you see the hand, if you think about it here, it starts to rotate on you. When we actually dive into the elbow and the mechanics of elbow flexion, you'll understand that a little bit better, but it'll kind of come full circle for you. And then here in D and E, you see how most of this force is straight out through the joint, thus causing that dislocating force. It's not as strong anymore. So, right here. So that's where I'll actually end the video for now, and we'll pick back up tomorrow with another video to help get you through this. Since we did spend 51 minutes here, that's probably enough. We'll probably have another 30 to 45-minute video tomorrow going in a little bit more depth on more mechanics of how muscles actually move bones. All right, so hope everyone has a good night. Take care, and see you tomorrow.